Embryology is a fascinating field that delves into the intricate development of various organ systems within the human body. Among these is the autonomic nervous system, a critical component responsible for regulating involuntary bodily functions. In this comprehensive script, we will explore the embryonic development of the autonomic nervous system, focusing on the genetic regulation of neural crest cell migration, lineage specification, ganglia formation, neurotransmitter involvement, maturation processes, neurotrophic factors, neuroendocrine regulation, and the clinical implications of developmental disorders. During embryonic development, neural crest cells originate in the ectoderm and migrate extensively throughout the embryo, eventually contributing to the formation of various tissues, including the autonomic nervous system. This migration is precisely regulated by specific genes and signaling pathways. Through complex interactions, transcription factors such as SOX10 and PAX3 control the expression of adhesion molecules, growth factors, and chemokine receptors, guiding neural crest cells towards their intended destinations within the embryo. Once neural crest cells have migrated to their appropriate locations, they undergo lineage specification, differentiating into distinct lineages within the autonomic nervous system. Two main divisions of the autonomic nervous system include the sympathetic and parasympathetic divisions. The differentiation process is mediated by a combination of intrinsic genetic programs and extrinsic signals received from the surrounding microenvironment. In the head region, various cranial autonomic ganglia form as part of the autonomic nervous system. These ganglia play essential roles in controlling functions such as salivation, pupillary constriction, and facial expressions. The development of these cranial ganglia involves the interaction of neural crest cells with local tissues, including the ectoderm and mesoderm-derived structures. Sympathetic preganglionic neurons, originating in the spinal cord, project to distant target tissues via sympathetic ganglia. During development, these neurons differentiate and extend their axons towards their target tissues through a process involving the guidance of axon growth cones along specific pathways. Signaling molecules, such as semaphorins and their receptors, play crucial roles in directing axon pathfinding. Neurotransmitters, such as acetylcholine and norepinephrine, play pivotal roles in guiding the differentiation and maturation of autonomic neurons. These signaling molecules act through specific receptors and trigger intracellular signaling cascades that influence neuronal survival, migration, axon growth, and synapse formation. The timing and spatial distribution of neurotransmitter expression are crucial for the proper development and function of the autonomic nervous system. The adrenal medulla, which shares common origins with sympathetic ganglia, plays a crucial role in the stress response by secreting hormones, including adrenaline. During embryonic development, neural crest cells give rise to both sympathetic neurons and chromaffin cells within the adrenal medulla. The interactions between these cell types and their surrounding microenvironment are necessary for the proper differentiation and function of the adrenal medulla. The growth and guidance of axons from autonomic neurons to their specific target tissues rely on complex molecular signaling pathways. Guidance cues, such as netrins, semaphorins, and ephrins, interact with their corresponding receptors on growth cones, triggering intracellular cascades that regulate axon growth, guidance, and target selection. The precise spatial and temporal control of these signaling events ensures accurate axon innervation of target tissues. The enteric nervous system plays a vital role in controlling gastrointestinal functions, including peristalsis and secretion. Neural crest cells contribute to the development of the enteric nervous system, forming ganglia and innervating the gut. Interactions between neural crest cells and the gut microenvironment, along with the intrinsic genetic programs, guide the proper formation and innervation of the enteric nervous system. Once neural crest cells have differentiated into post-ganglionic neurons and formed ganglia, further maturation processes take place. Synapses are formed between pre- and post-ganglionic neurons, establishing functional neural circuits. Neurotrophic factors, such as brain-derived neurotrophic factor, BDNF, and neurotrophins, play critical roles in promoting neuronal survival, axon growth, synapse formation, and the establishment of functional connections within autonomic ganglia. Neurotrophic factors, including nerve growth factor, NGF, brain-derived neurotrophic factor, BDNF, and neurotrophin 3, NT3, exert significant influences on autonomic nervous system development. These factors promote the survival, proliferation, migration, and differentiation of autonomic neurons, ensuring proper target tissue innervation and functional maturation. Imbalances in neurotrophic factor signaling can lead to developmental abnormalities within the autonomic nervous system. The autonomic ganglia regulate endocrine responses in various organs. Neuroendocrine factors, including hormones, growth factors, and peptides, influence the development and function of autonomic ganglia. Crosstalk between autonomic neurons and endocrine cells is critical for proper organ development, homeostasis, and the integration of physiological responses to internal and external stimuli. Understanding the embryonic development of the autonomic nervous system is crucial for comprehending the etiology and pathogenesis of clinical conditions such as autonomic dysfunction. Disorders like Horner's syndrome, dysautonomia, and familial dysautonomia can result from abnormalities in autonomic nervous system development. By studying these developmental disorders, we can gain insights into potential diagnostic markers, therapeutic approaches, and preventative strategies. In summary, the development of the autonomic nervous system involves intricate processes such as cell migration, ganglia formation, and neurotransmitter involvement. Understanding these processes not only helps us understand normal development but also offers potential treatments for clinical conditions.